because of how essential thiamine is to macronutrient metabolism, especially glucose metabolism, and when considering how the brain depends on glucose as its primary energy source, it comes as absolutely no surprise that thiamine plays a major role in neurological function. In fact, signs of thiamine deficiency are mainly neurological in nature. This is because thiamine plays a role in nerve function and the electrical activity of many tissues. Actually, thiamine triphosphate, a form that's not commonly viewed as active, it can donate one of its 3 phosphate groups to activate a protein called rhabsin. Rhabsin plays a very important role in what is called the neuromuscular junction. This is where nerve fibers coming from motor neurons in the brainstem and spinal cord connect with skeletal muscle to initiate muscle contractions using the neurotransmitter acetylcholine. Neurotransmitters, for those that don't know, are specialized molecules that neurons in different parts of the body use to communicate with one another or target tissues. The rhabsin protein essentially helps to anchor acetylcholine receptors at the neuromuscular junction, specifically on the muscles. These receptors are needed so that when acetylcholine is released by nerve fibers, it can bind to these receptors on muscle and enable muscle contraction. Thiamine donates phosphate groups for other synaptic proteins directly in the brain as well. Interestingly though, while the brain is vulnerable to thiamine depletion, and when I say vulnerable, I mean that reduced thiamine in the brain would have serious consequences, but the brain does not store substantial amounts of thiamine in it. However, different animal-based studies have shown that during periods of low thiamine intake, the brain is depleted of thiamine more slowly than other tissues. On the flip side, when therapeutic doses of thiamine are given, like in a repletion protocol, the brain isn't repleted as rapidly as other tissues like the liver, for example. This suggests that the brain keeps some degree of what we call homeostatic control over its thiamine status. Remember just a bit ago I mentioned that under conditions of insufficient thiamine, different metabolic pathways might upregulate themselves to help sustain energy production. One such pathway in the brain is known as the GABA shunt. GABA is the most abundant inhibitory neurotransmitter in the central nervous system, but that pathway through which it's produced can also help to yield ATP under certain circumstances. When not enough thiamine is present to fuel alpha-ketoglutarate dehydrogenase, this enzyme is unable to convert alpha-ketoglutarate into succinyl-CoA within the Krebs cycle. When alpha-ketoglutarate builds up, the GABA shunt pathway might increase its activity. This takes alpha-ketoglutarate and then converts it into glutamate, which is an excitatory neurotransmitter. This is further converted into GABA, and then it's able to make its conversion into succinate and then re-enter the Krebs cycle. So under conditions of thiamine deprivation, the GABA shunt pathway may upregulate to help yield energy. This phenomenon may actually explain why anorexic behavior is noted as a common characteristic of thiamine deficiency, more specifically subclinical thiamine deficiency. GABA activity increasing through this pathway in certain parts of the brain has been shown to inhibit feeding in animal models, and this has been tied to reduced appetite and eating observed in subclinical thiamine deficiency in humans. In fact, a loss of appetite is one of the earliest signs and symptoms of thiamine deficiency.